welcome everybody. We have the great honor to have Dr. Marie Davis with us today to talk about investigating the role of GBA and the propagation of Lewy bodies in Parkinson's. And hopefully we'll have about 10 minutes of Q&A. Dr. Davis is a movement disorder neurologist and researcher at the VA Puget Sound Healthcare System and assistant professor in the Department of Neurology at UW. And her research focuses on understanding mechanisms underlying the production and spread of pathogenic protein aggregation in aging and aging-related neurodegenerative diseases, in particular Parkinson's disease. Her group uses both Drosophila and cultured neurons and glia derived from induced pluripotent stem cells from patients to understand mechanisms leading to neurodegeneration that could be targeted for novel disease-modifying therapies. Dr. Davis received her BA in biology from Bryn Mawr College and MD PhD from New York University School of Medicine. She did internship in internal medicine and neurology residency here at University of Washington, Seattle, and movement disorder fellowship also here in Seattle at the VA Puget Sound Patrick, and did postdoctoral research in, I'm afraid I'm going to mispronounce his name, Dr. Leo Palenque's laboratory at the University of Washington. So I'll hand it over to Dr. Davis. We're very excited to hear the talk.
Great, thank you. Thanks so much, Jenny. Thanks for inviting me. As Jenny mentioned, I'm a movement disorder neurologist nearby and at the VA and UW, and my research has really focused on trying to understand how Parkinson's disease and other neurodegenerative diseases develop through the lens of looking at this one gene, GBA. So I'll be telling you more about that. So as you know, many neurodegenerative diseases are characterized by, by protein aggregates. And this is just summarizing some of those protein aggregates in some of the main neurodegenerative diseases. So in Parkinson's, it's characterized by Lewy bodies, where alpha-synuclein is a major component. Alzheimer's, we recognize amyloid plaques due to amyloid beta peptide aggregation and phosphorylated tau in the tangles. Huntington's disease has protein aggregates that contain mutant Huntington protein, and ALS also has very characteristic protein aggregates. But not only are there very specific protein aggregates within the neurons of each of these diseases, there's also a stereotypic temporospatial progression of or spread of these protein aggregates throughout the nervous system. And this spread correlates with the disease progression clinically. And so these first two columns of pictures of brains show the spread of progression of the plaques and tangles in Alzheimer's disease. And then this last column is showing the progression of Lewy body pathology, starting in the lower brainstem, moving up through the midbrain, and then eventually throughout the neocortex. And so I'm going to focus on Parkinson's disease because I'm a movement disorder neurologist, but I'm hoping that eventually what I talk about today can be hopefully applied in some way to other neurodegenerative diseases characterized by protein aggregates. So as many of you know, Parkinson's disease is the second most common neurodegenerative disease after Alzheimer's. And it's characterized, we now recognize, by both motor and non-motor symptoms, especially for non-motor cognitive decline. And then as I said before, the characteristic, the neuropathologic characteristic is Lewy bodies, which are protein-rich aggregates within the neurons. And those contain alpha-synuclein as well as ubiquitinated proteins. So proteins that are tagged with ubiquitin, and that is targeting those proteins for degradation inside the cell. And I didn't mention, feel free to ask any questions. I'm not sure if you're able to, anyway, I feel free to just interrupt me and ask any questions. So most Parkinson's disease cases are what we could call, we would call idiopathic or sporadic. There's no clear single genetic or environmental cause that we can point to. But we are learning more about the genetic influences that are a lot more complex than what we initially thought. So there is a handful of genes that can cause, that clearly cause Parkinson's disease. So if you have a mutation or two mutations within both alleles, you are expected to develop Parkinson's disease. And some of these genes can cause Parkinson's very early in life, in childhood, such as Parkin. However, all of these single gene causes of Parkinson's disease are very rare. In total, they probably account for less than 10% of all Parkinson's cases. So they have a very strong effect. They're strongly penetrant, but they're very rare. We've now become aware of a lot of common variants that have a small effect in increasing risk of Parkinson's disease, and those were identified in genome-wide association studies. But at this point, they're really not clinically relevant. Their effect is so small. So they have very weak penetrance, but they are very common in the general population. And so spanning this very large spectrum of genetic influences is GBA. So GBA is common. It's about 5 to 10% of Parkinson's patients actually carry a GBA mutation. And it does have a clinically relevant effect. So it increases the risk of developing Parkinson's disease by about 5-fold if you carry a mutation compared to someone who does not carry a mutation. So it really is kind of right in the middle of this spectrum of genetic influences. So that's made it a very interesting gene for trying to understand the pathogenesis of Parkinson's disease. So we already know quite a bit about GBA because recessive mutations cause Gaucher's disease. So that's the, Gaucher's is the most common lysosomal storage disease. But in Parkinson's, what we're looking at are actually heterozygous GBA mutations. So these are otherwise asymptomatic carriers who have an increased risk of developing Parkinson's. And more recently, over the past 5 to 10 years, we've become aware that GBA carriers not only have an increased risk, but when they do develop Parkinson's, they have a much more rapid progression of the disease symptoms and a faster decline in function. And so that's been shown in both motor symptoms and cognitive symptoms that there's a faster decline. So what is GBA? It encodes an enzyme that functions in the lysosome. So I know many of you don't usually think about cells and so the lysosome is an organelle that within cells that functions to degrade proteins and some other organelles within the cell. Often the proteins that are targeted for degradation in the lysosome are tagged with ubiquitin, and so that's going to come up later on. So GBA encodes this enzyme that breaks down a glycolipid, glucosylceramide. And it removes glucose from that molecule and results in glucose and ceramide. So ceramide, you probably recognize, is a very important molecule for many lipids, especially lipids that are in cell membranes and enriched in neuronal membranes. Excuse me. I've been interested recently in wondering whether GBA could actually be accelerating the propagation of Lewy pathology throughout the nervous system, and whether that might be why GBA carriers have a faster disease progression. So to address this, I've been using two model systems, and I'll tell you about both today. So the first is a fruit fly model, or Drosophila, and a second is neuronal, human neuronal cultures that are derived from a patient. So where we obtained some tissue from the patient, reprogrammed it to a more stem cell-like form, and then differentiated those cells into neurons. So first, before I talk more about the fruit fly model, you're probably wondering why we would even use flies. They're so different from humans. So one thing is that flies actually have a lot of the same genes as humans, but as homologs. So actually 75% of human genes causing disease are present, do have homologs in flies. There's a lot of genetic tools and reagents that are publicly available in flies. And flies are an organism that have complex behaviors, and so we can examine phenotypes looking at their ability to move, their behavior, and even cognition. And then lastly, they're really easy to raise. So they have a pretty short lifespan in the lab. They live about 3 months maximum, and you can grow lots of them really quickly. And so that really makes it a lot easier to look at aging-related phenotypes compared to the mouse models that take much longer to develop those phenotypes. So fruit flies do have a homolog to the human GBA gene, and that fruit fly gene has about 50% similarity in protein sequence to the human GBA gene, and it's expressed throughout the fruit fly, including the nervous system. So I'm going to just briefly summarize what we've found so far in our fly GBA deficient model. Oh, sorry, I forgot to mention our model for GBA deficiency is actually a deletion that removes most of the coding sequence of the fruit fly homolog for GBA. So flies that are homozygous for this deletion actually have a lot of similarities to the human disease characteristics. So similar to in human Parkinson's disease, patients are fruit flies that are deficient in GBA have decreased enzyme activity. They have a shortened lifespan. So the GBA deficient fruit flies live about 1 month on average, and the normal fruit flies will live about 3 months. So it's a pretty significant decrease in lifespan. The fruit flies actually have a locomotor deficit, so I'll show that, especially for movement disorder neurologists watching. Be interested to see that. And then they actually do have a memory deficit. So I'm happy to talk about that afterwards if you're interested in knowing exactly how we assay that. They do have evidence of neurodegeneration, and they have accelerated protein aggregation. So just for the interest of showing how fruit flies could actually have a movement disorder. So fruit flies normally always want to climb up against gravity. So if you tap them in a small tube where they really can't fly, and you tap them to the bottom, they will immediately start climbing up the walls of the tube against gravity. And then this is an example. These are not actually GBA mutant flies, but they're flies that are mutant for Parkin, which is another genetic cause of Parkinson's disease. You can see that they climb up much, much more slowly. So the GBA mutant flies are in blue here, and the controls are in red. And you can see that over time, as the flies get older, their climbing ability does decline normally, but the GBA mutant flies have a much more rapid decline in climbing ability. The other phenotype I want to highlight for you that I have mentioned already is the accelerated protein aggregation phenotype, which we think is somewhat analogous to Lewy body formation. So I'm just showing you this Western blot only because it's a pretty dramatic change between the controls and GBA mutants. I know most of you are not used to looking at this, but this is just showing how we assay for proteins that are tagged with ubiquitin and meant for degradation. We're looking at the soluble fraction and then a detergent insoluble fraction, which we think would represent really aggregated proteins, somewhat similar to Lewy bodies. So in normal control flies, there are a lot of ubiquitinated proteins in the soluble fraction, but not very much in the insoluble. And in our GBA mutant flies, you can see there's a really dramatic increase in both the soluble and insoluble fractions. And that is quantified here in red. This time are the GBA mutant flies. You can see there's a really dramatic increase compared to the controls. And when we put back the wild type gene in our GBA mutant flies here in green, it rescues that increased protein aggregation. So pretty much back to normal. And then when we actually look at aggregated protein in whole brains of flies up here, you can see that there's a dramatic increase in the GBA mutant flies. And also when we look at the muscle in the thorax, these are control flies, and these are the GBA mutant flies. Again, you can see a dramatic increase. So what I've showed you so far is there is a significant increase in protein aggregation in our GBA mutant flies that are deficient in GBA. And so that really is consistent with our clinically with what we see. But now we wanted to see, do GBA mutations accelerate the disease progression in our fly model in some way, or facilitate the spread from cell to cell of these protein aggregates? So the way we did this was we wanted to know if protein aggregation in one part of the fly, such as the brain, could be rescued by expressing the wild type GBA in a tissue outside of the brain, such as the muscle. So basically we looked at brain tissue to look for protein aggregation and see, put back in a GBA deficient fly, wild type GBA, just in the muscle, and see if there's rescue or decreased protein aggregation in the brain, or no rescue, and that continued increased protein aggregation. So what we found was there is a rescue in between tissues. So when we express that wild-type GBA gene back into GBA deficient flies in the muscle, we look just at the muscle and we do see a rescue in green. But also when we looked at the heads in these animals, we saw that same rescue. So that was a really big surprise to us. And when we actually looked at the protein aggregates in whole brains, so these are control flies, these are the GBA deficient flies, where we see that increased protein aggregation. We do see that there's a rescue, and these are GBA mutant flies where wild-type GBA is expressed just in the muscle. So we want to see the opposite. What if we express wild-type GBA in the GBA deficient flies just in the neuronal tissue? Would we see a rescue in the brain as well as muscle? And so this is showing that when we do look at the head in these animals, there is rescue again, so in green. And then we look at the muscle, we see that rescue as well. So it is going both ways. So we wondered what could be the mechanism of this rescue between tissues in the fly for protein aggregation. And there have been several possible mechanisms that have been posited. So one is direct connections between cells through what are called tunneling nanotubes. And then there's direct communication and transfer of proteins. Another is the cell may directly extrude our proteins right into the extracellular matrix through exocytosis. And then also cells can package proteins in vesicles, small vesicles called exosomes, and those are secreted and then taken up by recipient cells. So we ended up finding basically, I'll tell you the punchline, that we found evidence that this rescue between tissues that we're seeing in our GBA mutant flies may be mediated by exosomes. And the way that we found that was a little bit roundabout. So we ended up doing proteomic profiling of our flies. And we expected to see a pattern of protein, a pattern that would be consistent with really impaired protein degradation. Because as I've showed you already, our GBA mutant flies have significantly increased ubiquitinated proteins. And so this really suggests that there's some major problem with protein degradation. And so this is just showing what we expected to see. So each dot would represent an individual protein that's measured in our proteomic profiling. And on this axis, it's from faster protein turnover to slower turnover. And on this axis, from decreased abundance to increased abundance. So if there is a problem with protein degradation, we'd expect that most of the proteins would fall in this quadrant of slower turnover and increased abundance. And that's what we have seen with other flies that have mutations in genes really required for protein degradation. However, in GBA flies, we did not find that at all. And so that was a big surprise to us. What we actually found was a significant amount of the proteins were in this lower quadrant of increased abundance and faster turnover. And there were actually only 15% of proteins in the quadrant we were expecting to see most of the proteins of slower turnover and increased abundance. And when we looked at the proteins that were within this lower quadrant of faster turnover and increased abundance, they were highly enriched in proteins involved in machinery that's involved in exosome production. And so this is making a very long story short, but basically our proteomic profiling really suggested that there was something going on that was dysregulated in exosome production. And so we looked more closely at this, and this is just showing that the proteins that are involved in exosomes in either production or cargo are increased in abundance and also in faster turnover compared to the other proteins that were measured in our GBA mutant flies. And then proteins that are thought to be really sort of intrinsic to exosomes, such as RAB11 and ALEX, were specifically increased in exosomes that were isolated from our GBA mutant flies. So this is showing right here in exosomes compared to the rest of the fly, that RAB11 and ALEX proteins that are really thought to be kind of characteristic of exosomes was increased. And then we tried to actually measure if there was an increase in abundance of exosomes, and did find that although the size of the exosomes in our GBA mutant flies wasn't significantly different from controls, it was increased in abundance compared to the controls. So I'm going to kind of skip over a lot of the data to just kind of give you the overall punchline for what we found. What we found is that our GBA mutant flies have significantly increased protein aggregation. And when we look at these GBA mutant flies and just put back wild-type GBA in one part of the body, we can see that it can rescue the protein aggregation in another part. So when we put it back in the muscle, it can rescue protein aggregation in the muscle as well as the brain, and vice versa. And then I haven't showed you this, but it also rescues those changes that we saw in exosomes. So the increased levels of proteins intrinsic to exosomes in our GBA mutant flies are also back to normal. So GBA is affecting somehow exosomes. And so that really led us to hypothesize that GBA may be normally restricting protein aggregation throughout the organism, and GBA mutations are somehow leading to faster spread of protein aggregates. And so now we're really trying to figure out how GBA mutations are affecting the spread of protein aggregates through exosomes. And at this point, this is where this is our current work, and we're trying to figure this out. But we have a lot of models. So could there be excessive exosome release, or is GBA changing the contents of exosomes? And that is what we're really, I think that is something that we're looking at, are whether there's an increased alpha-synuclein and ubiquitinated proteins within these exosomes, and also how are they taken up by recipient cells. So far I've told you all the work so far that has been done in the fruit fly model, but fruit flies they have some limitations. Their neurons are extremely small. They're we can't really culture them in a dish to really look inside of them and look at these organelles very closely. And so I've developed a human neuronal model to try to complement that. And that's using the induced pluripotent stem cells from Parkinson's patients who carry a GBA mutation. And so in collaboration with the University of Washington Stem Cell and Regenerative Medicine Institute, I was able to make or generate iPSCs from a patient carrying a GBA mutation who has Parkinson's disease. We were also able to make a isogenic control. So basically we reverted this mutation using CRISPR-Cas9 gene editing back to wild type, but everything else should be the same. And so that's why we're calling it an isogenic GBA wild type control, as well as an age and sex matched healthy control iPSCs. So this is just showing you that we are able to make neurons that express markers consistent with mature neurons from our derived from our iPSCs from our GBA PD patient. And we do see a dramatic increase in ubiquitinated proteins in the neurons derived from the GBA PD iPSCs compared to controls, very similarly to what we see in the fruit fly model. So again, this is a Western blot, but all I just want to highlight is that there is this really dramatic increase in ubiquitinated proteins in the 3 clones we derived from the GBA PD patient compared to the isogenic GBA wild type control and an unrelated healthy control. And so right now we're really trying to look at what could be the basis for changes in what could be the basis for these changes in protein aggregation. And also we're looking at exosome production in these neurons. And exosomes, just briefly, are produced through this endovesicular trafficking process. Contents are kind of trafficked in these vesicles in the cell called the endosome. They eventually end up within small vesicles within a larger vesicle called the multivesicular body. And normally, this multivesicular body can either go back to the cells, fuse with the cell membrane and release these inner vesicles as exosomes, but it can also fuse with the lysosome, the organelle that I mentioned before that is responsible for degradation. And so what we think right now is that GBA, when there's a mutation or it's deficient, this process of the multivesicular body fusing with the lysosome for degradation may be blocked or decreased, and that is leading to an increase in protein aggregation within the cell, and perhaps an increase in trafficking of this multivesicular body to the cell membrane to release even more exosomes that may be containing some of these pathogenic protein aggregates, and that could be leading to the faster spread that we see in GBA deficiency. So I hope that in the future I'll be able to give you, show you the results of this experiment, but right now I just want to tell you the plan is that we're characterizing right now the exosomes that are produced by the neurons differentiated from iPSCs from GBA PD patient as well as healthy controls. And then hoping to eventually basically transfer those vesicles or exosomes to either GBA PD or control neurons and look at the uptake of those exosomes and whether they are causing faster propagation of protein aggregates in those recipient cells. So looking at whether it causes cell death in those recipient cells, if those recipient cells, because of GBA mutation, are having more difficulty or alterations in trafficking to the lysosome, and if they're causing increased aggregation within the recipient cells. And so just to summarize, we think that GBA mutations are increasing risk and accelerating progression of Parkinson's disease by promoting and initiating, promoting faster initiation and spread of protein aggregation via exosomes. And so, yeah, as I said, we're really trying to look now at the process of exosome formation and how GBA is influencing that. Is it influencing its morphology of exosomes, of the protein cargo, and also the uptake by recipient cells. So just to conclude, I hope, I've told you a lot of specific things about GBA and how it might be influencing from the cellular level to the cell-to-cell level. But basically, the main take-home points I hope is to convey is that GBA mutations increase the risk of developing Parkinson's disease, and they accelerate the progression of the disease. So we hope that understanding how GBA is influencing these processes could reveal some novel therapeutic targets eventually to slow the progression and spread in Parkinson's, as well as other aggregation, other neurodegenerative diseases that are characterized by protein aggregation. So I just want to thank a bunch of people. So my lab, which really only started a few years ago, we're at the VA, we're pretty small still, but we're growing. And my postdoc mentor, where this project really started, and I still collaborate with them, with Leo Polank's lab. I collaborate a lot with Jessica Young's lab at UW Ice Cream, or the Institute for Stem Cell and Regenerative Medicine, as well as their stem cell core, which is where we generated the iPSCs. And then hopefully I'll tell you more about this other fly model I've been working on. But anyway, so I'm happy to take questions.
It was a great talk, Dr. Davis. Thank you so much. Jenny, did you want to tee up some of the questions for her in the chat? Looks like we have 3.
Yeah, sure. Thank you so much, Marie. That was an amazing talk, and I'm such an admirer of the work you've put in all these years on trying to understand these pathways. So we have a question from Praveen Kamani. Thank you. Great talk, Dr. Davis. Is it true that baclofen and other GABA B agonists improve upward movement in fruit fly models of PD? If so, is there a role for GABA B drugs in PD? Thanks.
Oh, I'm actually not familiar with baclofen being given to fruit fly models, so I'll have to look into that. I mean, I know levodopa has been given to fruit flies, and that has shown benefit in certain phenotypes, but I wasn't aware of baclofen, so I'll have to look at that. So yeah, please send that to me because I'm not aware.
Okay, and then let's see, there was a question from Dr. Lopez. Genetic Parkinson's could be treated like this? that question came in at some point during your talk, and I'm not sure exactly what slide it was on, but maybe the questions pertain to, would potential therapeutic targets and that you're studying here be applicable to Parkinson's in general, or just GBA carriers?
Yeah. So I know there are specific therapies being targeted for specific, even specific GBA mutations, and that's and the good thing is GBA is not uncommon. As I mentioned, it's present in about 5 to 10% of PD patients, but that's still pretty rare, overall for a person just, for neurologists treating Parkinson's patients. And those therapies, I think, are really targeting the protein degradation pathways. And I didn't really talk about this, but even the genetics of GBA are kind of complex, because not all GBA mutations are thought to be equal. Some mutations are thought to maybe cause protein misfolding and lead to problems with just the production of the protein in the endoplasmic reticulum and getting stuck in there. Whereas other mutations are thought to really affect more the lysosome with protein degradation. And so there are kind of there's efforts to kind of target those two different processes for those different types of GBA mutations. But I guess I'm hoping that looking at how GBA is influencing the spread of protein aggregates from cell to cell could be more applicable in general, because we do know that GBA carriers in general have faster progression, and so this does seem to be a more common kind of process that's affected, rather than looking specifically within the cell. Is it affecting this process versus this process? So I think the end result is there is faster progression, and that is something we can try to target. And the fact that GBA seems to be influencing cell-to-cell propagation, possibly through exosomes, I think could be potentially a really exciting target. So that is why I'm hoping it'll also be applicable to other neurodegenerative diseases as well, because we may not be able to completely stop the disease, but if we can halt or slow that progression, I think that would be really great. So I don't know if that answers the question, but it brings up more questions.
We require a lot of crystal balling. Let's see. Dr. Kamani had another question here. Also, any role for, and I'm not sure how to pronounce this because I'm not familiar with this thing either, but miglustat in GBA carriers?
Yeah, miglustat. Yeah, so miglustat is used in Gaucher's and also in Niemann-Pick type C, and it's basically it inhibits the formation of glucosylceramide. So yeah, it has been really thought as a potentially a possible therapy. And the one issue is that miglustat is not thought to really be able to cross the blood-brain barrier very well. And so I know that there are efforts to try to make a better form of that drug to cross the blood-brain barrier. And that has been looked at in animal models and there were some promising results from that. But right now it's not something we usually do or give. But I think that is something that could be really promising.
Okay, and then it looks like there's a question from Denise Russom, who's our clinic supervisor. Do these mutations influence Lewy body accumulations in the fruit fly models?
Yeah. So we can't totally say that fruit flies have Lewy bodies, but we call them these aggregates that we think are somewhat similar. And one of the things I didn't mention is that fruit flies actually don't have alpha-synuclein. They just don't have that protein. But when we express the human alpha-synuclein in our GBA mutant flies, there is increased oligomerization or aggregation of alpha-synuclein in the GBA mutant flies. So the protein aggregation is definitely, loss of function or decreased function of GBA is definitely promoting protein aggregation even independently of alpha-synuclein in our fruit fly model.
So the proteins that you're seeing increased aggregation in are just any proteins. They could be tau, they could be amyloid, they could be anything.
Yeah, so we haven't ever really tried to look at exactly what proteins because it's tough with these insoluble aggregates to break them up to do proteomics. But those Western blots I showed, they're just a big smear, and that's because they're all different sizes. So that smear is going from big proteins to small proteins, and you can just see it's just across the board, kind of a big smear of proteins. So yeah, there's all sorts of proteins that are tagged with ubiquitin that are staying in the tissue, in the cells. And yeah, so it's a whole range that would be targeted for degradation.
That's wow, that's amazing. So I wonder why in humans carriers are more specifically at risk for Parkinson's as a neurodegenerative disease as opposed to any neurodegenerative proteinopathy.
Yeah. I think that's a major question.
Yeah.
Well, so actually, so GBA carriers, I really didn't talk about this, but they also have a very increased risk of Lewy body dementia. So, and so I guess I kind of simplified it just as, just for Parkinson, but they have an even higher risk, like an 8-fold risk of developing Lewy body dementia compared to non-carriers. But again, those are synucleinopathies, not tauopathies or Alzheimer's. You're absolutely right. It is.
Does it matter which type of GBA mutation, whether they have more of the Parkinson's disease phenotype or the Lewy body phenotype?
I don't think we've had any correlation, between GBA, specific GBA mutations and Lewy body dementia versus PD. Yeah, I don't think that's been seen so far.
Is there any relationship to MSA?
So it does not look like that's borne out. Yeah, people have looked at that to see if because MSA is basically Lewy bodies within glia or astrocytes, and that does not seem to be the case, which is interesting. Yeah. So yeah, when I didn't really talk about this, but my plan also is to look at astrocytes, not just neurons. So with a lot of these neurodegenerative diseases, so far we've really focused on neuronal pathology and kind of put aside any glial influences. And so I think with the iPSC technology, it allows us to differentiate, different cell types, including astrocytes and microglia, which we're now recognizing more and more that the immune function as well as support from astrocytes in clearing protein aggregates might also play a really big role. So yeah, I'm hoping to look at those cell types as well.
Very cool. You might have covered this, and I apologize if I'm taking over all the question time. So other people, please chime in if you have questions in the chat or Q&A box. But you might have covered this, and it just went over my head. But what is the mechanism that the wild type GBA could rescue a distant organ that carries the mutation? How is that thought to happen?
Yeah, so at this point, we think it's through somehow influencing exosomes, but we're not 100% sure. So in that the data that really supports that that really keyed us into that mechanism was initially through the proteomic profiling that kind of hinted towards that. It showed that there was this dysregulation with proteins involved in exosome formation. And then we specifically looked at exosomes after that in our fly model. And so we actually have looked at knockdown of some genes involved in exosome formation, and they do seem to influence the protein aggregation phenotype and that rescue between tissues. So it's a little hard to pin down because a lot of the genes involved in exosome formation are very vital genes, and so when we really knock them down in expression, it kills the fly, or we don't knock it down enough, it just doesn't really have much of an effect. And so that's why I'm really starting to look more in the cell culture model. Also just because it's easier to look, into the neurons individually and see, what's going on, because the fruit fly neurons are just, they're too small.
And this is also maybe too much of a, distant future question, but maybe other people are wondering the same thing. But if you can rescue a fruit fly model with this wild type expression of GBA, do you think in the future there would be any kind of gene therapy role for this in humans to do the same kind of rescue?
Yeah, and people are thinking about that a lot, actually. So, and GBA is not only, so we see a decrease in enzyme function because GBA encodes an enzyme. We see a decrease in enzyme function not only in Parkinson's patients who carry a GBA mutation, but also in Parkinson's patients who have normal GBA. So it is affected even in I guess there's like some funneling of the whole disease process that does still end up affecting GBA enzyme activity. And so, yeah, I think it could not only be applicable to GBA carriers, but potentially, non-carriers with the disease.
I mean, that would be amazing if that could be applied in an organ that's not the brain too, because
Yeah.
you do these surgical vector implants.
Yeah.
Apply it to the gut or something, maybe. I don't know.
Right.
It does make your imagination kind of go wild about like, oh, it's possible. It's amazing.
Right.
Okay. Let's see. Any other questions in the box? I don't see any. Are you still looking for patients to donate cells at all? Do you need us to send folks who have familial Parkinson's to you?
Yeah, absolutely. So, so the person to talk to would be Cyrus Dabedian, because I collaborate closely with him, and he has the whole biorepository and everything set up. And then that is actually through him that I got the tissue to make the iPSCs. So yeah, absolutely, we're always interested. And actually, so far I've really just been working with neurons derived from iPSCs from this one patient, but I eventually do want to have more than one for sure. So.
Well, that's awesome. Thank you so much for this amazing talk and sharing your work. It's really exciting. I guess just for the rest of the audience, it looks like Megan put in the chat box how to claim CME credit. So it looks like we have 2 hours after the conference to text code 144 to 1-844-989-1333. And then the evaluation is at the SSF Cloud -cme.com to finish claiming credit. Thank you very much, Dr. Davis, and we hope to have you back in the future to hear how things have developed and hear the rest of the story.
Yeah, great. Thanks for having me.
Thanks for watching. Hit the subscribe button for more medical content.